Hello and welcome to our video lecture on section 7.6 on the connections of carbohydrate, protein, and lipid metabolic pathways. We know that these pathways are going to be interconnected and will create lots of different types of intermediates, and what they can do is leave to join other pathways. Note that these pathways are not closed systems, so a lot of the substrates, intermediates, and products are going to be reactants in other pathways. Our learning objectives here are going to be to discuss the ways in which carbohydrate, metabolic pathways, glycolysis, and the citric acid cycle interrelate with protein and lipid metabolic pathways, and explain why metabolic pathways are not considered closed systems. So we'll start off talking about the connections of other sugars to glucose metabolism. All of our catabolic pathways for carbs, proteins, and lipids are going to eventually lead and connect to glycolysis and the citric acid pathways. So we'll start off with glycogen, and we've talked about glycogen before in this chapter and probably in other chapters too, and it is a polymer of glucose, meaning that it is a large molecule that is going to contain these repeated glucose molecules that are all linked together, and it is a way to store energy in animals. We can store this within our liver and our muscle cells, and so what we can do is hydrolyze it into glucose 1-phosphate monomers if we have low blood sugar, and then from there we can convert it into glucose 6-phosphate. And that should sound familiar to you because that is going to be one of those first steps within our glycolytic pathway, within glycolysis. And so if we didn't have low blood glucose, we would of course just take the glucose we already have within our blood. But anytime we see that we have low blood glucose, we're going to call upon glycogen to supply the glucose. Now, on the other side of this, if we have excess glucose, then we are going to take those glucose molecules and bind them together, create glycogen, and store that in the liver and the muscle. We can also note that by having glycogen in storage allows us to produce ATP for a longer period of time during exercise. Other ways we can gain or utilize sugars is to use fructose and galactose. These are both monosaccharides in order to make ATP the same amount as glucose. And then we can also catabolize sucrose, which is a disaccharide. We can see here glucose is connected with a glycosidic bond to fructose in order to make ATP as well. Now let's talk about the connections of proteins to glucose metabolism. When we have excess amino acids or our body starts to go into starvation mode, we can shunt amino acids into our glucose catabolic pathways. Now this isn't something we want to do. Usually we're going to use our amino acids in order to make new proteins within our body, but like we said, if we have an excess amount or we have no other sources of fuel, we will have to take that amino acid and remove the amino group, and then it can enter the pathway. What happens to that amino group though? It is going to be turned into ammonia, and in the liver we'll take 2 ammonia molecules and convert it into urea, which is going to be a waste product that we find within the urine. And we have our gas of carbon dioxide that is left over. Now note that we have amino acids that are entering this process in many different places. So all in the red here are the different types of amino acids that we have. And maybe you recognize what this circle here is. This is our citric acid cycle. And so depending on what type of amino acid we have will determine in what area or what step it is going to be able to enter in on. Now our connections of lipid and glucose metabolisms. We know that grouped within the lipid umbrella, we have cholesterol and triglycerides. cholesterol, if you remember from our cell membrane or plasma membrane lectures, it's going to be found within there to allow for flexibility, and also is going to serve as a precursor for our steroid hormones. And our triglycerides, if you remember, was a bonding of one glycerol, and we had 3 fatty acids. And this is a really great way of our body to be able to store some long-term energy. And we take that glycerol and we can phosphorylate it, meaning we're going to add a phosphate to it, and we get glycerol 3-phosphate that can then enter the glycolysis pathway. With our fatty acids, we are going to be able to catabolize that into a process known as beta oxidation. And this process takes place in the mitochondrial matrix, as well as the Krebs cycle and electron chain transport. And we have our 2 units of acetyl groups picked up by our coenzyme A that turns it into acetyl-CoA. Now, if you remember, and I'm going to go back here to remind you, we have acetyl-CoA that is going to enter the Krebs cycle and allow us to move down the pathway. And this is just a nice summary slide for you to see where all of these macromolecules are playing roles within in order to allow cellular respiration to continue.